Okay, good morning. Any questions before we get going? I hope I got YouTube set up so that it doesn't start playing as I start talking. Seems good. So real quickly, there is a a few practice questions that have been posted. It's all just some true false, but it covers each chapter, a handful of questions to kind of get you thinking a little bit about main idea. There is summary posted, there's extra credit stuff posted, and there will be very soon, can't guarantee that it will be today, but I'm hoping, I will get a review set up for you guys that kind of goes through more details, or a study guide, whatever you want to call it. Not just summary stuff that will hopefully be a little more tailored toward the exam than just the study guide that's posted. That's just really just a simple review that's on there. So I will get that. I have had a handful of questions or requests for something like that, so I will try to get something like that put together soon. It is, for me, considerably easier to do that once the test has all been put together. Then I can go through and truly tailor that for you. And I got that all completed over the weekend. So that should be posted on Moodle. It'll likely be posted right under next week's material. That's where the test is and all of that stuff. On the extra credit essay, I'm assuming you should be able to submit that. I don't know why it's telling you it's incomplete. Go ahead and submit it, and I will get on there a little later today and check to see what your submission is. If it's good, no problems. If not, I'll let you know. So it should be. And I think if you're submitting it as a file, it's got to be like a Word document or a PDF, I believe. So make sure that it's proper doc or PDF, whatever it is. I think that might be it. Okay. Let's see. I thought there was 2 chats. I think that it was all on the same one. Okay. So if there are no other questions, we will get started. There's 2 chapters left. This one, the focus is really the adaptive or the humoral, the antibody-mediated types of a response. As I mentioned before, we often think as these as separate types of interactions. Part of that is true, but really we think about a lot of this interacting together, where you've got more than just one type of a response that's trying to target whatever types of foreign invaders that we're thinking about. So even though we talk about these as separate actions, they're oftentimes working together, okay? So there are lots of details as we move through here about how these mechanisms work. And as we wrap this unit up in chapter 19, we're going to talk about some issues, some problems when the immune response kind of goes haywire. We'll talk about potential for different autoimmune diseases and other situations where, the potential for organisms to maybe eliminate or circumvent the responses that we're trying to mount against them. More of that will kind of hit in the last unit of material when we think about some of the specific pathogens and some of their strategies. But with that, we can at least kind of introduce some of those thoughts as we wrap up this unit. So again, we're thinking kind of more antibody approach as we move through this chunk of material. This first figure is something that, it's more historical account. We don't usually think about iron lungs. We don't probably think about polio because it's something that is kind of out of sight, out of mind. We don't see issues with it in the United States. We have almost eliminated this. It's something that we're on the cusp of saying that it's no longer an issue. It still is a problem, but it kind of really depends on where you're at. So this was a relatively common situation as we see this polio ward that's basically just full of these devices. They're called iron lungs. And basically, one of the issues with polio was the potential for paralysis, and in these circumstances, severe paralysis to the point that they're unable to breathe on their own. So they're basically surviving in these iron lungs where it's doing the job of breathing. We've got other potentials, ventilators and things like that, that kind of take over these circumstances. From what I know, there in the United States, there is one gentleman that still utilizes an iron lung. Not something that we really think about. Do you have a question? It's, I think, yes, they are. I think they are kind of like a hyperbaric chamber. I think they're, the idea with that, I mean, it's putting pressure, the hyperbaric chamber is pressurized and it's helping to increase the oxygen that's really being forced into you. This, I think, is similar to that. I'm not exactly 100% with it, but I know that it's altering pressure that in turn helps to force the muscles to make you breathe. So kind of the same sort of an idea. So when we think about an immune response, an antibody response, this is a classical picture of kind of what we might expect to see happening. And it depends on the circumstances. It depends on the specific things that our body's mounting a response against, as far as how pronounced these responses might be, as well as how the longevity of these responses. The time down here, this is all just arbitrary. We could be thinking months, we could push it out maybe thinking potentially years with this. It sort of just depends. Concentration up here, also, we don't have any real scale on this at this point, all right? But we could make this quantifiable. We could do tests that measure serum concentrations of antibody over the course of time and plot this to get this type of a response, right? And know specific levels or quantities of antibody in that serum specimen. We would call that a titer. How much of that are we dealing with, right? And it's a mechanism when we think about serology, when we think about trying to detect specific substances like antibodies in serum specimens. It's a mechanism that allows us to monitor, to gauge how our body is doing with their respective response against these pathogens. So we can use it as a monitoring device. We can use it to determine if a person's actually mounting any sort of a response against this antibody-wise, right? So there are, and again, this kind of just an arbitrary image. We'll see another image of this probably Wednesday. We will see another image that shows different classes of antibodies and kind of their trend over the course of, the same sort of an idea. And it sort of mirrors the same image. So initially with this, we start off with our initial exposure. This could be coming in contact with some foreign invader, or we could think about it like a vaccination situation, and it could be the first dosage of that vaccine, the first encounter with whatever foreign substance. This is our initial exposure. And we could say this is time zero. This is the first instance, all right? And then the clock starts ticking. And usually when we think about this first bit on the x-axis, we'd be counting it off in days, usually seeing a primary immune response between a week or two. So 7 to 14 days, you start seeing, definitely measurable amounts of antibody that you could detect in that serum specimen. The concentration on this primary response compared to the second dosage is much less, all right? And we'll hit that here in just a second. So again, over the course of time, that level of antibody is going to peak and then it will start to decline. This typically, again, thinking days or so, maybe weeks, maybe at about 30 days or about a month into it, we see it starting to decline. And again, there's different classes of immunoglobulins, different classes of antibodies, where these bars or this line will alter the littlest bit, all right? But still, it starts to decline. You can see there's still measurable amounts, but it's less than this peak, all right? Here's the secondary exposure. So again, depends on what we're talking about. It could be you got a vaccination on that initial exposure, and the secondary exposure is the real player, or it's possibly a booster shot. So often vaccination schedules require more than one. It depends on the circumstances, right? Uh, Regardless, the second exposure, whatever it might be, we come in contact with the same player, if you will, and now this response is much more pronounced. And uh, at this point, still we can be talking days, but what maybe took 2 weeks to start seeing really this peak sort of response, you get to that same sort of a level right here within a day or two after the secondary exposure. It's right at that same level of that primary response, almost instantaneously. As it sees this second exposure, the antibody production is basically kicked into gear. It's with memory cells that were still kind of laying in wait. They are prepared to start producing this antibody almost instantaneously. So you see a response very rapidly, and within a handful of days, it is towering well above what that primary response was. So you get a serious response based off of this. And these immune cells, after they've initially been stimulated to be produced, upon that secondary exposure, they mount a much more dramatic response against this foreign invader. And if it truly is the bad guy, well, that's a good situation because now you've got massive amounts of these antibodies released into the circulation to basically be targeting these foreign invaders. If it's the booster shot, it provides that extra level of protection as you mount a bigger response. So it stays more pronounced and for a longer duration. Usually we see this, although it looks like maybe we're talking about the same amount. As it starts to decline, those levels remain considerably more elevated even than that initial primary response. This depends on the type of substance. Some things we may see this second response, or after this booster shot, we may see these levels remaining elevated almost indefinitely, thinking lifelong immunity. There are other potentials where you'll see it decline and requiring booster shots later on, or possibly requiring a whole new round of vaccination, dependent upon the circumstance, as it would drop off. Things like the flu vaccine, we've got different proteins expressed on the surface of these viruses that cause the need for different types of vaccines every year. And you would expect the secondary response, or the response really, because you're only getting one shot, but you would expect that initial response to decline and dropping to levels that ultimately you're going to need something again, even if And it's highly unlikely, but even if it's the same strain of influenza the following year, a booster shot or a new vaccine would be warranted because your antibody levels would definitely have declined by that point. So, primary, secondary immune response, again, this is kind of the classical situation where you expect to see a measurable, a pretty good level for that primary response, and then much more in that secondary exposure, that secondary immune response, right? And again, it kind of depends on the circumstances. It depends on what it is that we're mounting a response against in the first place, as far as how much concentration-wise, how much we're producing, and the duration of that as well is going to definitely vary, right? We could think about an allergic reaction as an example while we've got this up here, right? We will kind of think more about it a little bit later. But in the situation with allergies, there's all sorts of potentials with this. And there are things that you may say, you know, well, I was born with this, maybe. There are definitely genetic situations that set you up for some of these. There are allergies that you can contract when you're young and you grow out of them. Or there are allergies that you get later in life. You didn't have it before and now you do, all right? It really just kind of depends, all right? We're not gonna go off and think about all of those different things at this point. But ultimately what happens in types of allergic reactions are you need something kind of like this figure, right? Something where you get an initial exposure to this foreign substance, whatever it might be, all right? And we call that initial exposure a sensitizing dose. This is where you get the first little hint of this thing, and your body has the potential to produce all sorts of different types of antibodies that will stimulate different types of reactions, right? And the allergic situation is kind of a problem, all right? It's not so good if you're dealing with these allergies. It's your body's mechanism of saying, this thing's foreign and I don't like it, and we're going to deal with it, right? So, specific class of immunoglobulin gets tasked, gets produced in this response, and it sets the stage for preparing us to respond unfavorably, allergic reaction like, all right? So this sensitizing dose initiates these cells within our body to say, okay, this is maybe a bad guy. And the next exposure, whatever it might be, all right, is what we call the shocking dose. So you first get sensitized, then your body comes in contact with it again, and now it's shocked. Now you respond, all right? Now you get a serious allergic reaction, all right? And those cells that had kind of been laying in wait are kicked into overdrive very rapidly, and the response is very pronounced, right? There's numerous things that can fit with this, all right? One, and this is a little bit different as we're thinking about these types of responses, but it's a similar sort of an idea, all right? Poison ivy, things like that. It is a type of a reaction, contact dermatitis, but it's not really thinking the same sort of an idea with antibodies. It's a cell-mediated response, right? But there's still similar ideas with memory cells and seeing it upon the next go-around, right? So you may come in contact and you may not recognize anything. And usually you're a little kid when this happens, all right? And you brush up against some poison ivy, no issues. And then later, it happens again. And this is when you actually really get the reaction. And with things like that, being a cell-mediated response, and this is really the next chapter of material, but as we see this right here, it's fine to talk about and think about it a little bit. That response takes time, right? So it could be a situation where a day or two goes by before your skin really starts to experience these situations. Again, with it's different than the antibody, but it's the same sort of an idea. All right. So our body responds to all sorts of foreign players, and it responds differently dependent upon what those foreign players are, how they present themselves, if you will. So this antigen is a molecule, potentially a protein molecule. Those are really good antigens. Doesn't have to just be protein, but protein's pretty good, all right? So you've got some macromolecule, some big glob of something that our body can detect, and maybe it says you're all fine and good, or maybe it sees it and says, I don't like the way you look, right? There's something about you that's maybe just not right, and we want to respond accordingly, all right? We respond by looking at this antigen and really looking at its specific fingerprint, if you will, all right? The epitopes are specific regions on this antigen, thinking if it's a protein, we could think of these respective epitopes as a handful of amino acids that have a specific characteristic on that protein, and it is something that may be recognized by some immune cells and recognized as being pretty foreign, being something that's maybe hazardous, right? So we see these epitopes and detect them, mount a response accordingly. And on a specific antigen, as we've got right here, there's the potential that it may have multiple types of epitopes, multiple little strands of amino acid, chains of amino acids that give it a specific characteristic. And as we look at this one antigen, 3 specific things that make it stand out as being pretty stinking foreign is really what we want to think about, all right? The more foreign you are, the more likely we are to produce a pretty pronounced response. If you are very, very similar to our own types of cells, or your epitopes look similar to our own sorts of molecular fingerprints, well, then you would hope that the response would not be expressed at all, right? You don't want to respond to something that looks like your own stuff. Then we start thinking about some adverse reactions and potential autoimmune situations when we target our own stuff, maybe based off of something that we saw before. That's an issue, right? So here we've got 3 different looking epitopes, 3 different potentials on this antigen to recognize and mount varied types of responses, or for that matter, to produce different antibodies, all from this one antigen, all right? 3 different types of immunoglobulins could be created to recognize, you know, the pyramid, the spherical bit, or this little rectangular fragment, and we'll respond accordingly. So this is same sort of an idea, and each of these little Y-looking guys in here are antibodies, and we've got antibodies that are specific to their respective epitopes, right? So greens, yellows, and reds, right, for their respective shapes that we're looking at. And each one of these antigens, with its varied epitopes being presented, have the potential for these antibodies to recognize and link up with them, all right? In this case, these antibodies, these immunoglobulins, they're snagging multiple epitopes on different antigens, and it causes these substances, whatever it might be, this could be a bacterium with specific proteins on the outside of it, and we're snagging the whole thing. Or it could be a molecule that's in your circulation, some soluble protein moving through the body, a toxin that's been secreted by one of these foreign invaders. Antibodies might recognize that as well, right? Whatever it is, the foreign substance with its respective epitopes gets snagged by these antibodies. And this image that we're seeing is called an agglutination reaction. It's the clumping of these foreign molecules basically pulling them out, taking them out of commission so that they're no longer just kind of free floating around causing damage, right? This may lend to other cells recognizing this clump and coming in and trying to respond. There's other potentials where this portion of the immunoglobulin, the little tail, can stimulate immune cells as well. We'll hit that here in a second, right? There's multiple ways that this all acts to try to remove the foreign obstacles from our system in the first place. So there's all sorts of variation when we think about antibodies and we think about the respective epitopes. There are numerous things that are potentially recognized as being foreign. And there's a lot of stuff when we think about in our own body, there's a lot of stuff that we need to recognize as either being our own, or we need to recognize that you're not a bad guy in the first place and you're okay. We don't want to mount a response against you, right? So this becomes super important when we think about trying to control these types of responses and trying to ensure that the response doesn't target ourselves, right? So we'll hit more of that in a little bit. So this is a generalized image of an immunoglobulin, of an antibody. And there are, like I said, multiple classes of antibodies. They have this, but they may potentially have handfuls of them or two of them together. Just kind of depends. So antibodies are proteins, all right? And they are multiple chains of proteins linked together. So we've got this heavy chain, which is in purple, right? And it makes a Y. And there is a light chain over here, these green guys, right, on the sides. And they make half of the top bit of the Y, right? So heavy and light chain. And on the top, this FAB, it's called, well, part of the FAB is the variable region. It's the antigen binding site, FAB, for fragment antigen binding. And this variable region, you can kind of see here, we've made it look kind of like a shape in here. It's sort of like an enzyme and its substrate, like a lock and key, where you've got specificity for this. It's really kind of the same idea. And the antigen binding site is specific for whatever epitope, the circles and triangles and whatever we showed before. That little sequence of amino acid has a specific three-dimensional shape, and the antigen binding site has the same sort of shape to allow them to link up together, right? Specifically, right? For the most part, right? There are some issues where these sites may look sort of similar and you might get a little bit of cross-linking, but that again is kind of an extreme, right? We think of these as being highly specific for that respective epitope, all right? So the variable region is what's going to do the business of linking up with the specific epitopes, right? And then poking out would be this FC region, the two heavy chain tails, and this is a crystallizable fragment. So there are, that's the FC for crystallizable fragment. There are all sorts of techniques where we can crystallize structures, crystallizing proteins or nucleic acids, and then bombarding them with X-ray and seeing the diffraction patterns. That's really what we're talking about with this, is looking at it through X-ray diffraction. This bit, well, does that very well. Here's a ribbon model of the immunoglobulin, and you can see these interconnecting bits as kind of alpha helices, and then the beta pleated sheets. giving it kind of these bits right here, the structure, if you will. Light and heavy chain linked together, and the two are held together by disulfide bonds, a pair of cysteines adjacent to one another that cross-link and covalently bond, holding them tight. So again, there are variations with the respective types or classes of immunoglobulins, but they all have this generalized idea with binding site, cross-linking, and at least one of these, right? So this one right here is just a monomer, a single one linked together. And this, let's see, let me stop this, see if I can bring it to this, maybe. That's not, I don't think that's what I'm sharing. Okay. All right, I'm assuming you are, those of you tuning in at home are seeing this table and it's actually visible. That's what we're seeing here. So let me move this over the littlest bit. So this, let me tell you what table it is. Table 18.6. It is in the PowerPoints, but it is a little bit blurry. So with this, here are the different classes of immunoglobulins. So there's 5 different types. They've got some different potentials as far as what they might be doing. There are some bits in here that you don't need to think about, but there are a few that are definitely important as we're thinking about understanding a little bit of the variation. So IG for immunoglobulin, and then the other bit, so for its specific one that we're looking at, and it's based off of their heavy chains that we're looking at as they've got some of these designations. So IgG exists as a monomer, a single Y. IgM is a pentamer. There are 5 immunoglobulins linked together, creating the potential for it to interact with considerably more antigens, more players, because we've got multiple binding sites. So thinking agglutination reactions, IgM is a great one for that. The next one over is IgA. It's a, well, it's here we've got secretory dimer. So 2 of the immunoglobulins linked together. IgA can exist as both monomer and dimer, so as a single Y or as the double. And with that, in the dimer form, we see this in lots of mucus secretions. IgA is very important. It helps to prevent infection by interacting with substances that might come in first contact with the innate response. And as it comes in contact with these mucus, the antibody has the potential to recognize and link up to it, coating it and preventing it from attaching in the first place. IgD is a monomer, and so is IgE. So it's the M and the A that have some variation, right? Pentamer and dimer, respectively. Their binding sites, so the antigens that they've got the capability of linking up with, are varied. Monomers can only bind 2, so 2 binding sites. The pentamer has the potential to link up with 10, and our dimer 4, right? The weights of these guys kind of makes sense that the pentamer and dimer would be considerably larger than the respective monomers. So as far as their concentration goes, IgG is the dominant immunoglobulin, upwards of 80% of this uh, immunoglobulin, if we were just looking at antibodies within the serum, 80% would be IgG, then IgM, and the rest relatively low. The secretory stuff, about 13%. And in the serum, it's existing as the monomer. In mucus is when you would see the dimer form. So IgG is important, and that's this column right here. IgG is important because it's the only immunoglobulin that is able to cross the placenta. So it imparts immunity to that unborn child. You can also see it present in breast milk. So a breastfeeding child is picking up immunoglobulin that way from its mother as well. So it can cross the placenta, providing immunity, providing protective mechanisms that are essential. It also has the capability of fixing complement. So it participates in the complement cascade, in the classical pathway of complement fixation, initiating these proteins in the circulation to start destroying these foreign invaders. IgM also has the capability of doing that. So these two, as far as immune recognition, stimulating complement, they have a huge role in that. The Fc bit on the IgG, and again, it's the dominant form in the serum. It can link up to phagocytes, so macrophages or neutrophils can recognize the Fc region of the IgG immunoglobulin, and it can cause those cells, it can cause their destruction. The back up here, IgE, sorry, over here also is important. The Fc region of it can link up to mast cells and to basophils, all right? So IgE is the one that I was mentioning, thinking about an allergic reaction, an allergic response, all right? You oftentimes think about the bad guys, these foreign invaders, and it's IgM and IgG that are the immunoglobulins produced against these foreign invaders to basically deal with this substance, whatever it might be. But if IgE happens to be produced in these circumstances, when it comes in contact with these foreign invaders, it stimulates other things, not the macrophages, but mast cells and basophils. And these cells have histamine, which in turn is an issue. And we start thinking allergic reactions as the histamine is released from these cells, causing causing inflammation, causing the symptoms, the runny nose, watery eyes, all sorts of issues with that, and it's histamine that's really the culprit with that. So their functions down below are really kind of the importance with this, as well as does it cross the placenta, does it fix complement? These are the bits that you really kind of want to think about with these immunoglobulins. So IgG and IgM can neutralize substances, so they can link up to maybe toxins, taking those toxins out of commission as they bind to them. They can cause agglutination, what we showed with that image with those different antigen, those epitopes getting linked up with immunoglobulins. They can fix complement or activate the complement to cause lysis. They can tag things by opsonizing, so sticking to them. The IgG does this by opsonizing, and it's that Fc region that's sticking out, kind of like a flag. It's like walking through the forest and seeing some trees that have tags on them. They're going to come through and maybe cut them out because they're diseased or something like that. That little flag indicating something's up. The Fc region on the IgG molecule is like that. So these cells circulating through, or these immunoglobulins moving through, linking up to cells, stimulates our immune cells, like macrophages, as they're doing surveillance to see the substance has a tag on it. It's got a flag. we need to destroy. They're also important with antibody-dependent cell-mediated cytotoxicity. So stimulating other cells, like a natural killer cell, as an example, to recognize kind of still sort of the opsonization. It sees this Fc region, but it recognizes there's something up here, I'm going to destroy it, all right? So IgG, a big player in the immune response, and it's got multiple modes of action to really try to enhance the capability of destroying these from any angle that we can. So we also see with IgM not only the activation of complement, but it can serve as a B cell receptor. So it can stimulate, basically communicating directly with those B cells, which are going to be important for the production of antibodies, right? So that's a big player with that. This guy right here, the IgA, with neutralization and trapping pathogens within the mucus, as we're thinking secretory. The IgD is a B cell receptor, so it helps with immune communication and regulation, antibody production, as far as that goes. And then the IgE, again, kind of bad situation, as we see activation of basophils and mast cells. So allergic reactions and potential parasitic infection, right? So that may warrant, and I think we mentioned this before, it may potentially warrant some further investigation. Eosinophils, type of granulocyte, are important in parasitic infections. So too are the IgE molecules. Seeing both of these elevated stimulates some thinking that maybe that's what's going on, but you also deep down need to think it could just simply be hay fever as the potential. Okay, let me stop that. I think this is the exact same one. Okay. So this is where we just dealt with this. So here, with the neutralization, there's a couple of different things. Kind of depends on the circumstances as far as what we might be doing. So this, in this case, we've got our IgG molecule. Here's a virus, and we're linking up to some of these spikes on this viral molecule that basically make it so it's unable to dock with or to link up to its respective host cell that wants it, that the virus wants to infect. So neutralizing it prevents it from attaching. Over here, the diphtheria toxin, so it's not the bacteria, but the toxin that the bacteria is producing, there's antibodies against that, and it can link up to specific areas of the toxin that, again, would want to link up to a host cell, to that receptor, and it blocks it, makes it so it doesn't fit into that receptor, and it can't act, basically inactivating those toxins. Over here, we've linked up to the flagella on the bacterium. So multiple potentials as far as neutralizing goes, blocking their activity, preventing their attachment or docking, that basically prevents this from being progressed through to actually establish infection. So as far as opsonization goes, the Fc regions act like that little flag, if you will, coating the cell, stimulating, coating the pathogen. I mean, it can be an intact cell. It could be fragments of it, but it coats this and stimulates a macrophage to destroy. So the macrophage has all sorts of potential receptors on its surface that allow it to really do its job of surveillance. And these green guys are Fc receptors. So they're moving around through the tissues, searching for foreign substances and searching for antibodies that are serving as the little flags to say we found something bad in the first place. So the antibody links up to epitopes on this pathogen, through its FAB region, so the top of the Y, docks with specific epitopes, and that FC region can link up to the FC receptor on the macrophage, causing the macrophage to engulf, to invaginate, to pull in this pathogen, and we will destroy this bacteria as a result of this opsonization, right? This coating. So there are numerous potentials with this as far as being able to agglutinate, to pull things out. IgM, as I said a second ago, the fact that it's a pentamer, so 10 binding sites, makes it really good at being able to grab these foreign invaders and cause them to clump together. So it binds to multiple cells, has the potential to do that, and that causes, again, this agglutination. And these are bacterial cells, I'm assuming, but if this, since they're red, we could assume maybe these were erythrocytes, you could have a transfusion reaction where you're given the wrong blood, and you can have antibodies in your body that will recognize this stuff and cause these erythrocytes to do just this, to clump up. That's hemagglutination when we talk about red cells, and a potential serious or fatal blood clot could be the result of this reaction. And it kind of, depends on how much you were transfused with, but there's definitely some potential serious interactions with that. Okay, we'll pick up with the antibody-dependent cell-mediated stuff. We'll start with this on Wednesday. Any questions before I quit? Okay, guys, like I said, I will get a review posted very soon, so be looking for that on Moodle. Farewell. Have a good day.